we have someone with substantial experience and leadership in government and the private sector to speak on the overview of the translational research landscape to begin our workshop. Dr. Christopher Austin is the director of the National Center for Advancing Translational Sciences at the National Institutes of Health and has had that position since 2012. Before joining the NIH in 2002, he led several drug development programs at Merck. From 2016 to 2018, he served as chair of the International Rare Disease Research Consortium, and he is a member of the National Academy of Medicine. So please join me in welcoming Dr. Austin for his presentation, and then we will have some time for questions. Dr. Austin, thank you for coming.
Well, thank you, Senator Bingaman, and it's great to be here. This is something that I and my colleagues think about a lot, and what I'm going to try to do is give you just what my assignment was, to give you a landscape. I may give you a few opinions and perspectives in there someplace, but what I really want to do is set the landscape for what promises to be some very rich discussions. Before I do that, I need to reiterate that that NIH as a whole, of course, and NCATS in particular, agrees that affordability of drugs is an issue of concern. And this, as such, this issue is being addressed by our partner, our parent organization, the Department of Health and Human Services, which has developed a blueprint that we hope will help to bring down prescription drug prices. But NIH's mission as a part of HHS is, of course, to focus on conducting and supporting the research that will eventually lead to these interventions. And we don't have a role in drug pricing. One of the distinctions that I'll make is between price, which we don't have any role in, and cost of production, which we very much hope to impact. And I'll talk about that a little bit. And as we know, cost of production and price are inexactly related to each other. But it's certainly, from our point of view, the cost of production, which I'll talk about, is a huge issue in this space, and will allow those prices to we hope become a little bit more manageable. So I'm just going to jump right in here. So. When I thought about the landscape, what I constantly find is that the landscape of drug development is even more than most areas of human endeavor, depending on the perspective of and the position within the ecosystem that one sits. And you remember this famous New Yorker cartoon that has the world being on 9th Avenue and maybe a little bit on 10th Avenue, and way in the distance there is the Hudson River, and then there's the Pacific Ocean and the rest of the world, and this is very much the way players in this very complicated space tend to see the world as well. They tend to have a keyhole view in the world, and I find that this leads to just lack of knowledge, which can lead to some erroneous conclusions sometimes, just as if one would think that the world really is all close to 9th Avenue, which of course it isn't, even though it might seem that way when you're there. And this ecosystem is very broad, and I'm going to use these icons or these as indicative of all of the people who are supported by these organizations. So it's not NIH as an organization, but it's all NIH employees and grantees, of which there are hundreds of thousands all over the country and the world doing research. And then in the academic world, there is actually something called the Academic Drug Discovery Consortium, and their point of view is a little different. This is from someone that some of you may know, Josh Summer, who is both a patient of has a disease called chordoma, but also is an entrepreneur, social entrepreneur, who runs his own foundation to cure his own disease before the disease kills him. So he has a particular point of view as well. And then the venture capital organizations in this country, which are really a national treasure, I think, that as I travel the world, most other countries do not have, and it's responsible for a lot of the dynamism in this space. Those venture capital folks and private equity have a particular point of view. And then, of course, the many thousands of biotech organizations that they're part of bio and not part of bio, and then the larger companies that make up pharma. And by, again, I don't mean the pharma organization. What I'm talking about is the pharma companies, because I couldn't fit all their logos in here. It'd be too busy a slide. FDA we'll hear from later. Janet Woodcock will be here, but they have a particular point of view. CMS, and again by CMS I mean payers, public and private payers. So I'm using them to be a sort of a representative of the payer community. And then the reason that we're all doing this is the patients in the first place. You might recognize Megan Crowley, who was at the State of the Union address a few years ago, suffers from Pompe disease, and her father, John Crowley, who started a company that developed the treatment that Megan is now on. And one of the things that we might want to talk about at some point is the role of patients in therapeutic development, which has really been a very, very positive development in our view. So the point being that depending on where you sit, you are going to view this problem in a different way. And I hope during the course of the next 2 days, we constantly put ourselves in the position of the other and realize that others have a different point of view that we do that may be equally valid, just different from ours. The other thing I run into frequently is that, at least in the academic space, there's an asymmetry of knowledge. So everybody in a pharmaceutical organization has at one time pretty much been in an academic organization. So they understand how academic organizations work, and they understand the role of academic organizations. But the reverse is not true. Most people in academia have never actually done most of the vast majority of what goes into making a new therapeutic. And so like me, who had a sort of revelatory experience, and it's a universal experience that you find when people go from academia to organizations that actually produce therapeutics, they have oh boy, I'm amazed at what I didn't know. And sometimes, myself included, will say, gosh, I can't believe I said all those stupid things that I said before I went to this organization. And it's not for any reason that people just haven't been exposed. They don't know the knowledge. So myself and others at NIH, including Amir Tamiz, who's sitting right there in the fifth or so row at the Neurological Institute at NIH, we spend a lot of time talking to our colleagues about this, and we'll talk about it today. So you probably all know of this concept called the Valley of Death, and I just want to touch on it for a very specific reason. The Valley of Death is used to describe the therapeutic development process. Now, if you knew nothing else, that should tell you that this is not something to engage in lightly, because you're probably not going to make it out the other end. That's the definition of a valley of death. So this should indicate to us extreme difficulty and likelihood that you're going to fail. That's why it's called the Valley of Death in the first place. And this cartoon is from Nature a few years ago, but it's meant to show the basic investigator on one side and the clinician and the patient on the other side. And who's on the bottom there, that's the therapeutic developer. And they're not looking too good. And that's where what happens to most attempts to make a therapeutic. And this is the problem that the center that I now run is focused on. Because until we understand and transform the Valley of Death into a predictable transversable process that will allow us to efficiently get from one side to the other, we're going to be left with inefficiencies and costs which are just unsustainable. So we view this as a scientific problem, and I think it's very clearly is a scientific problem, among other things. So this is a fundamental problem. So I think that the question is, and what I think we ought to be focused on, is how do we as a community eliminate the valley of death? The valley of death is not a law of physics. It's not a fundamental law of thermodynamics. It exists because we don't understand what the rules are by which we go from a fundamental discovery to a therapeutic that is shown to improve human health. Okay, so as part of another part of the National Academies, one of the other boards, part of the Division of Health and Medicine, there's a forum called the Forum for Drug Development and Translation, and a number of us, myself and Amir and others, Janet, are on that forum. Steve Golson was the co-chair of that until recently. And some of you, just to give you some names, Rob Califf is the current co-chair. So those are the kinds of people that's on this. And we realized a few years ago that though this group knew, because we all work in this space, that every participant in this very complicated team effort contributes to this very complicated process, which is replete with handoffs from one sector to the other. There was actually not a map to demonstrate to those who have never done this before what it actually looks like, and to help us actually re-engineer the process. And the problem you can see here that many stakeholders, probably most I think, are unaware of the complexity of what it really takes to develop a new therapeutic. And so we felt that we needed to create an accurate portrayal of the translational process, and this forum did that in a crowdsourcing kind of way. And then we published this in Nature Reviews Drug Discovery and in CTS a few years ago. And you'll notice, just you probably can't see this from the back, but the authors are indicative of how much of a team process this is. John Wagner is at Takeda, Andy Dahlem was at Lilly at the time. Lynn Hudson is at Critical Path, and she'll talk later. Sharon Terry is at the Genetic Alliance. She's a patient advocate. Russ Altman's at Stanford. Taylor Gilliland and myself and Chris DeFeo are all associated with NCATS, a government organization. And so we're all singing from the same songbook. And I want to show you what this was. And we started out with the realization that we were our own worst enemy in the terms that we use. I, up until recently, I will admit guilt on this, have used, like everyone else does, the term drug development pipeline. Now, what is a pipeline? Everybody knows what a pipeline is. You have them hopefully going to your house and carrying water. A pipe is a conduit into which an entity goes in one end and traverses the pipe and comes out the other end unchanged. And the progress through the pipe is dependent only on its diameter. Now, that is about as unindicative of the failure-prone process of therapeutic development as a word could possibly be. But this is how we tend to diagram it, and it looks really simple. And so what we find is that people actually believe us. And then we say, well, no, it's really hard. And they say, well, you just called it a pipeline. How hard could it be? It just goes from one end to the other. Water rolls downhill. And that's perfectly understandable because of the words that we use. And when you look at this, it does actually look quite simple. But what we did was to break this out and actually ask, well, what are all the interconnected parts? And this turned into this. And this is actually what it looks like. And this is a quite simplified version of this process. And whenever I talk to my fundamental science colleagues, and I'm actually a fundamental geneticist, I'm a model organism geneticist by training, so I started my career cloning genes and working on genomes and things. Those people and the Russ Altmans of the world, they fit up here in the upper left corner. And yet, given the New Yorker sort of analogy, they believe that's the whole world. And so what we needed to do is to show them that no, this is a very rich world up here, But if they actually want to make a drug, they got to get through all of this. And think about this as the board game from hell. You know, you will never, most people never get through this space. And there are recursive loops here that you can get stuck in, or things that are just impossible to do, as it turns out scientifically, or have to be done 100 or 1,000 times in order to get a success. And so we're using this actually and have created, using it for educational purposes, but also have created a Google Map version of this, because, you know, in Google Maps are useful because they show you where the trouble spots are. You know, where's the red, where's the traffic, and so we created a traffic version of this to show, you know, what are the areas that are greatest area of greatest trouble spots in terms of cost, failure, or time. And we've also created, for folks' use, that you might want to look at some point, a mobile version of this where you can zoom in and zoom out, and then we're adding programs that you might be able to go to for help in one of these spaces or another. I don't want to dwell on this except to say that this is the world that we're talking about here. And it's the current reality. Now, we can try to engineer this space, but this is the reality and that we're dealing with now. So what effect does this have? Well, it gives us the world that we all live in, which is, I find, a remarkably bittersweet time from the standpoint of being a scientist and a doctor, and I'm both. I'm a geneticist on one side and a neurologist on the other. And my science half is just in nirvana. You know, the things that scientists can do now compared to when I myself was in training 30, 35 years ago have been completely transformed. I mean, the productivity and the kinds of things that individual investigators can do was simply unimaginable, truly, 30 years ago in one professional lifetime. And this is perhaps exemplified by the Genome Project, and at the bottom those are induced pluripotent stem cells, which, remember, were only invented about 12 years ago, and those are differentiating into neurons and putting out nerve cell processes. But this was all science fiction when I was in training. And it's reality now. It's things that first-week graduate students sequence genomes and make iPS cells. People are doing this in high school now. But on the other side, you have to ask, well, how are we doing on the health side? And there's a wonderful report, depressing but good, a report from the Institute of Medicine, when it was still called that a few years ago, called Shorter Lives, Poorer Health. Sort of says it all. And this is a child with progeria. He actually died a couple of years after that at the age of about 15 of heart disease. Premature aging syndrome. So the point is that though we have these remarkable insights on the science side, the health side has not kept up. And to the degree that advances in health are dependent on advances in science, we have to ask ourselves, why is that? It's a curious observation. One would have expected that one would have followed from the other. And fundamentally, the reason is what we just talked about. There's a very poor transition from what's on the left side and the right side. But there is now an enormous, unprecedented opportunity to address these health problems that we never had before. And let me show you an example of this. There's lots of examples of this, but this is my current, one of my favorites. So just graphically, just sort of lays it out. So what you're looking at here is the number of human conditions, most of these are diseases, that have a known molecular basis, the gene for cystic fibrosis, Huntington's disease, sickle cell disease, and on and on and on. When I was in training, and when I graduated from medical school, off on the left there, I was going to say it was easy to study for the boards because there were only about 10 diseases, the molecular basis of which was known. And now, 30 years later, that number is about 6,500 diseases, the molecular basis of which we know. And now, just think about that for a second. For the whole history of scientific endeavor, we didn't understand the genetic causes of genetic diseases. And over the last 30 years, we now understand essentially all of them, at least the rare diseases and many of the common ones as well. That has completely transformed what science can do and how medicines how we can think about making medicines. The problem is, if you're a patient, beyond the diagnosis, this is what you run into. That only about 500 of these have any treatment. So 95% of human diseases have no treatment. And we'll come back to that. But you might say, well, gosh, you know, you haven't said this, but I will say it in a minute, that a drug takes, and you probably know these numbers, on average, currently, the current state of the art is about 15 years. And so you think, well, gosh, maybe if we just wait 15 years, there will be a time shift and the number of drugs will go up too. Over that period of time. And unfortunately, you would be wrong. And the reason you would be wrong is a derivative of this. Now, if you don't know what Moore's Law is, please don't embarrass yourself by admitting it. Google it after this. And Moore's Law will allow you to Google it. Moore's Law is the number of transistors you can fit on a microprocessor, which is named after Gordon Moore, one of the founders of Intel. It's this observation that the number of microprocessors you can fit on a transistor has doubled every year since 1970. The cost has come down contemporaneously during that time. That's why our world has been transformed by computing, and you could fit what was a supercomputer 30 years ago into an iPhone. But okay, so how are we doing during the same period of time in drug development, therapeutic development? Well, unfortunately, it looks like this. So this comes from a real paper a couple of years ago, and it's titled, it's called Eroom's Law. This is a little apocryphal, but this is Moore's Law spelled backwards. And this is a profoundly frightening graph to me for a number of reasons, and should be to all of you. So what you're looking at here is monotonically negative productivity growth in therapeutic development year after year since 1950, such that the number of new drugs approved per $1 billion spent has decreased by half every 9 years since 1950. Now, I don't care whether you're talking about shoes or ball bearings or drugs or cars. If you have an industry with this kind of productivity, negative productivity growth over 6 or 7 decades, what's going to happen? Those companies are going to have difficulty keeping going. They're going to merge with each other. They might go out of business, and the costs and the drugs that they do make are going to be, or the products that they do make are going to be exorbitantly expensive. Okay, again, I don't care whether you're talking about shoes or drugs or anything else. That's pure economics. So this, and think about the science that I just told you. So none of this has been affected by the invention of computers, recombinant DNA, gene therapy, the Human Genome Project, CRISPR. None of it has had any effect on this monotone. Some people think it's actually made it worse. Now, from our point of view, the solution to this is the same as what Matt Damon did on The Martian. You remember this when he was stuck in this? You remember, this is a family show. It's being broadcast, so I can't use the term he actually used. But what he said was, we are going to science the heck out of this problem when he got to Mars. And that's our solution to this. But I just want to lay the landscape for you that this is the reality that people who are working in this space currently work. And it's not because people aren't smart and committed and working their tails off night and day to not only make current drugs but make it better. It's just this is a very, very, very hard problem. Okay, so what the way I always put this to trainees, and I use these slides all the time, is that what I was taught when I was in academia actually isn't right. So when I was a neurologist, I was taught all I had to do was make a diagnosis and everything else was fine. And that's a patient with a stroke. And neurologists are famous for this. You know, we can exquisitely diagnose your stroke, but we can't do anything for you. And most patients are pretty dissatisfied with that. And geneticists are just as bad. You know, you'll find okay, you, Mrs. Jones, you have an A to T mutation, and that's causing your disease. Have a nice day. And I thought, not because anybody ever told me this, but because it was just it was implied that once I understood the diagnosis, when I had the diagnosis, and once I had characterized intellectually and scientifically the clinical syndrome or the genetic syndrome, then the therapy would just happen. You know, it's like the end of a lot of scientific papers. If you notice, it says, and there will be a drug in the next 3 to 5 years. That's the end of every scientific paper ends in 2 ways: either that or more research is needed. In other words, keep funding my lab. There's two endpoints. And so reading these, I and everybody else thought, well, okay, there will be a drug as the night follows the day. And unfortunately, that turned out to be wrong. So this is not equal to this. That's a device at the bottom. And what's more, though we probably won't talk about it today, but we spend an awful lot of time thinking about this, is that this is not equal to this. That is, we actually may talk about it. This is access. Do all the patients who could use this drug actually get it, and do they benefit or not? And the current numbers is it takes about 15 years and fails 99% of the time for the first inequality. And the second inequality, we don't have a failure rate as fine there, but the best numbers are it takes 17 years to go from the middle to the other side, even longer than it does to make the drug in the first place. So, and that's a health systems delivery dissemination problem. So these two inequalities, when you think about it, are the translational problem, and they're the problem that we work on and the science underlying that process. So I had to show this because you can't, whenever you're going to show a drug development slide, you always have to show this famous slide, and lots of people show it. This happens to come from pharma, but there's multiple versions of this, and they all show the same things. That is that this process has a tremendous amount of attrition to it, and it takes 10 to 15 years, costs, depending on how you do the math and whether you include cost of capital or not, somewhere around $2 to $10 billion, fails between 90 and 99% of the time, depending on when you start and when you stop. And if you think about this, and those of you who heard Andrew Lowe from MIT talk about this, he's quite eloquent talking about this, and he says, okay, so I have an investment possibility for you here. You can invest in this, and it's going to be 15 years before you know whether there's any result, and it's going to fail 99% of the time and cost $2 billion. When do you want to invest? Or you can invest in Facebook or the next Candy Crush, and 6 months from now you'll have a bunch of eyeballs on this, and your investment will have quadrupled in that period of time. So it's just, but this is, again, I paint this to give you my job is to show you what the landscape is, and this is the landscape. It's not a very satisfactory landscape, grant you, and a lot of smart people are working on this, but this is the landscape. Okay, so this is another way to put this. It's basically the same slide here, but with same picture, but with same diagram, but with pictures, and it's a simplified version. It's sometimes what I call drug development in 6 easy steps. But the point I want to make from this slide is something that I talk to academics about a lot who are getting into this space, and it's another really hard truth, which is if you divide this, just for the sake of this argument, from basic research, and I think we all know what that is, discovering a fundamental phenomenon, or in a normal system where when something goes wrong in a disease, why does it go wrong? From there to developing a tool, small molecule, sort of a proto-drug to test in cells or animals about whether your idea might be true, that stage, to going to a investigational drug that you could actually test in humans, to actually testing in humans. From the first stage to the second, to the third, to the fourth, the unit cost per project goes up by a log each time. So what you can get for a graduate student, $100,000 a year, if the next stage will cost you about $1 million per project, the next stage will cost you about $10 million per project, and a cheap clinical trial will cost you $100 million. And so as we at the NIH want to translate some of these discoveries on the left side to what's on the right side, the bill is not arithmetic or continuous, it's exponential. So sometimes we're in a Sophie's Choice kind of situation here, because if we want to do one discovery project, that's 10 postdocs we can't support to do fundamental research, or 100 postdocs to make one that will go to people. And so those are the numbers. And again, we're trying to make the numbers better. That's what NCATS does, increase efficiency, make this better, faster, more efficient. But again, this is the landscape that we're dealing with today. Another one, I just threw this in because this is a sort of industry standard paper. It was produced by Steve Paul just as he was leaving Eli Lilly. And this gives you numbers from Lily's perspective, and I'm not going to go through these in any detail. The numbers I would just want to show you here is their cumulative success rate here is 4%, and their total out-of-pocket cost is not capitalized now. Out-of-pocket cost was $873 million for one launch, and that's not counting all the failures. So these are the numbers. So almost $1 billion for one drug. Okay, so I hope you're not, if you're getting depressed, things are going to get better. So now, but that's the landscape, and it's important that we all understand what that landscape is. So I hope I've convinced you now that the landscape, we do not want to live in this current valley of death. We have an absolute societal imperative and scientific imperative and medical imperative to change this. And that's what my organization does. That's what parts of NIH, like the part that Amir runs, do. But it's really, this is all of those other organizations that I showed you at the very beginning with those logos. That's what they do. And so innovation and translation is really key to increasing efficiency. And the bang for your buck here is decreasing failure rate. Because if you go from a failure rate of 99% to 98%, you've doubled your success rate. And so we're focusing on failure. So again, the point here is not to tell you about NCATS. What I'm going to tell you about, the reason I'm going to tell you about NCATS is just to give you a flavor of, to look through our lens about this landscape. So we were started about 7 years ago with the purpose of addressing this problem, to bend the E-Rooms law curve, if you will. And so we're doing this by developing new ways of developing therapeutics better, faster, and more efficiently across all human diseases. And for the purposes of NCATS, we had to define what we mean by translation. What we really mean is observations into interventions. There's an observation a scientist might make in a lab, a doc might make in a clinic, a community health worker might make in the community, turning that observation, which may or may not be robust and reproducible, that observation into an actual thing, a drug, a device, a behavioral intervention, whatever it is, and showing that it actually works in individuals and the public. That's translation. But that's not translational science. Translational science is what we do, which is the field of investigation that seeks to understand what the rules are, so that if this process that I was just describing, which has this horrific failure rate and cost as a result of the failure rate, we need to, if we can succeed in transforming this whole process from a phenomenological trial and error empirical process, which is what it is now, into a predictive science, change it from what I sometimes feel it is now, which is astrology, to engineering. And my own field of genetics has done this over the last 50 years. That kind of transformation is what's needed. And now, of course, genetics is quite efficient and reproducible and effective now. And that's the kind of translation efficiency that we need as well. And so I show you this because these are some of the problems that we work on, and you notice that none of these are fundamental science, which is really the heart of what NIH does. And thank heaven that NIH does this. NIH is the world's biggest funder of biomedical research, and NIH is, without the NIH investment in fundamental research, we would have nothing to translate, and so we are absolutely dependent on our colleagues for that. But at NCATS, we don't work on fundamental science, at least not on purpose. These are all the other things that we work on, which are all the reasons that translational projects fail. I said it fails 99% of the time. Why? These are all the problems. You notice there's no disease names here because they're the same. Doesn't matter whether you're working on Alzheimer's disease or psoriasis. They're all the same problems. But they're not only scientific problems, they're organizational and cultural problems too, and we're addressing one of those today. Things like data transparency, IP management, credit for what is inherently a team exercise, education and training, scientific and cultural training, and collaborative structures, public-private structures, this handoff from the public sector to the private sector, because the private sector is the one that's going to make those therapeutics. NIH, with very, very, very rare exceptions, isn't going to do that. It's just not our mission. And so just very briefly, a couple things about how we do this. And again, don't worry about the details. I want to give you a sense of the way we're approaching this problem. About 70% of our effort goes into the clinical domain. Much of this is through something called the CTSA program that works on the bottom half of that problem list I showed you at about 60 institutions worldwide. You notice particularly the bottom one, the creation of an academic discipline of translational science. One of the things that we're doing is directly attacking the clinical inefficiencies through something called the Trial Innovation Network, which is a national laboratory to understand and innovate on the very inefficient process of studying an intervention in people, sort of a learning clinical trial system by analogy to the learning healthcare system, and we're developing and testing new processes to improve flexibility, speed, adaptive trial designs, those kinds of things, efficiency and quality, while reducing overall costs. And this is, we want to answer clinically important questions. That's really important. Clinically important questions that clinicians need to know and come in on time and on budget, which is actually a rare phenomenon currently, given how inefficient this process is. One of the things we've done is to solve something that was said to be impossible. Those of you who know about the Multisite IRB problem, 6-7 years ago, a typical multi-site trial with 20 or 30 sites, which is actually a relatively small trial now, could be delayed for 2 years in having all the lawyers at the individual institutions review the IRB, the Institutional Review Board agreement. And this was not adding any safety. It was just delaying for 2 years in time and cost, patients being able to get into clinical trials, and patients get sicker, become ineligible for clinical trials, or die waiting for us to make the IRB system work. It's just unconscionable. But this problem is now solved. Because NCATS did this in collaboration with all our collaborators at the CTSAs, and you probably can't see this from the back, but there's now 601 institutions across the country that have all signed this treaty where they agree to rely on each other, and one organization will do the IRB review and everybody else will rely on them. We've done the same thing on contracts. Instead of having to redo contracts every time, over and over and over again. These things sound trivial. They can delay a project for 2 years. And again, time equals lives, as far as Faster Cures likes to say. Patients become ineligible and die waiting for these human-made processes to happen. And we're addressing those. A huge problem in recruitment. In NIH-funded trials, upwards of 50% of them often end in futility because they never recruit enough patients to actually study in the trial. It's only about 5% of patients who have an illness who actually take part in clinical trials. It's quite remarkable. And so this is both a cultural problem, particularly our underrepresented groups who have typically not been well represented in clinical trials, and it's an informatics problem. So we've funded a recruitment innovation center at Vanderbilt and Meharry to do this, focused on not only community engagement and outreach, but also data-driven recruitment via extra electronic health records. So the reason I told you that is what are we actually doing? Is it actually possible to bend Eroom's law, to decrease costs, to increase efficiency? The answer is yes, but it requires a different way of working than what we've worked before. I want to give you a sense of the landscape of rare diseases because it's a particularly compelling issue and is becoming a major issue when it comes to cost. I mean, the new SMA gene therapy is $2.1 million per patient. So where are we with rare diseases? It's really a quite remarkable situation, landscape. There's thought to be about 7,000 of these. About 80% of them are genetic ones that you've heard about, like sickle cell and Huntington's and CF. Most of them are pediatric. And an interesting number that you probably don't know is that there's about 250 new rare diseases being defined every single year. So every other day there's a new rare disease which we have to treat. And this number, this increase is not plateauing. The slope is still increasing. So how many rare diseases there actually are, the jury's out on. I think the number is likely to be about 10,000. That's a lot. So even though they're individually rare, this number of diseases, they're cumulatively common. They make up about 8% of the population, which is about the same as type 2 diabetes. Everybody's heard of type 2 diabetes. many people, most people have never heard of a rare disease. They are responsible for enormous costs, 100 to sometimes 1,000-fold greater than age-matched control, especially in kids, in direct patient care, loss to productivity. Accurate diagnosis typically takes 5 to 15 years. It's known as the diagnostic odyssey. Huge healthcare costs incurred in this wandering in the wilderness trying to find a clinician who recognizes your disorder. And only about 5% of these have any treatment approved by the FDA. Only about 5%. These are the diseases that I used to see as a neurologist. Lots of pediatric neurodegenerative diseases, ALS, Huntington's disease, glioblastoma, what John McCain died of. These are all rare neurological disorders, and they are no more treatable now than they were when I was in training 30 years ago. And I did a calculation a few years ago, and I said, well, at the current rate of innovation, how many years will it be before every rare disease has a treatment? And the answer is 2,000 years. 2,000 years. Now, any Room's law will tell us that the price of a new drug will be infinity by that time. So we cannot continue doing what we're currently doing. And that's my lesson for you now. And so we have to think about how this ecosystem is going to remain not only healthy, but become much more healthy than it currently is, because it's got a big task in front of it. So what we're doing is relatively straightforward from a conceptual standpoint. It's difficult from a implementation standpoint, is to stop focusing on one disease at a time. These diseases are all related to each other. They've been considered 10,000 different diseases, but they're not. They're all connected to each other. We've just never been studying them that way. So going to a many diseases at a time approach, looking for commonalities among diseases, looking at platforms for development. We're doing this in a variety of ways, providing information to patients, big data to enable AI and other things, empowering patients directly as research partners, having patients as members of the research team from the very beginning on every project we do, developing registries which are interoperable, and doing lots of natural histories and interventional studies through the Rare Disease Clinical Research Network, which involves about 12 different NIH institutes and centers. We're also working a lot on development of gene therapy platforms. That is a sort of a cassette model. We're working with the FDA to find out, well, gosh, could we develop, a few AAVs, that maybe even ones that are approaching off patent, and do all of the tox and CMC and manufacturing and all that required to do, especially for very rare diseases, to allow many diseases to be tested simultaneously. And I just want to give you an example of why we're doing this, what's possible now. So this is a disease that I used to see when I was in practice. This is a pediatric neurological disease called aromatic amino acid decarboxylase deficiency, or AADC for short. This is a collaboration between NCATS and a company in Boston called Agilis. And once we reached a point of proof of concept, early proof of concept in people, then it was purchased by PTC Therapeutics, and they're going to do the definitive clinical trials and all of the manufacturing and CMC, all the work that's required to get this approved. But I just show you this. I don't know if you can see this from the back, but this little girl is about 4 years old in this picture, and this is a very typical pediatric neurological disease. She's barely responding to her parents' sort of grimaces in response to stimuli, and she would be downhill from there, would be dead long before the age of 10. This is a year after getting gene therapy into her brain, and this is her 2 years later in first grade. And if you watch this video, you would be hard-pressed to distinguish this child from any other child in first grade. This is what is now possible. And we have got to figure out a way to make this scalable. And that's so I just want to make sure we all understand why this is so important. Okay, so just lastly, I just want to tell you about one example of something we're doing focused on the question of why drugs fail so much in development. And you probably heard the number that even when a drug gets out of preclinical development, where it could be for 10 years and cost of hundreds of millions of dollars just to get through preclinical development, some at NIH often and some at a company, when they go into people, everybody gets excited, gosh, the drug's going to people for the first time, wonderful, but 90% of the time that drug is never approved. And why is that? Well, it turns out that about 60% of the time, the efficacy that was seen, the effectiveness that was seen in the animal model is not seen in a human. And about 30% of the time, it was safe in an animal, but not in a human. Ipso facto, rats are not humans. And sometimes we forget that at our peril. And if you look at the concordance between humans and animal toxicities, it's about 50% or less, sort of a flip of a coin. So one of the things that we've been asking, if this is a major cause of failure, we have got to address this problem. Animal testing for toxicology has not changed very much in 50 years. But could we, for instance, use IPS technology or tissue printing or spheroids made of human cells to be able to do identification of drugs in the first place. And so we're doing all of these things. The field is doing all of these things to try to address this problem. So we're focusing on complex human systems, not on animal systems. We've started doing bioprinting, like if you have a 3D printer in your house or in your lab, you can print plastics, but you can also print cells. So we're doing this in retina and blood vessel and skin and others. And this is something you may have heard about because it's had a big splash, kind of captures the imagination. These are microfluidic systems which combine stem cell technologies and tissue printing and microfluidics and cell sensors and imaging and computing to make little human microfluidic organs on a chip, in a microfluidic platform. And these are about the size of a microscope slide or maybe a little bit bigger. And they represent the structure and function of all human tissues. And we use those for studying safety and efficacy, not animals. And they're made from iPS cells for the most part, so we can make these for each person in this room. This was a pipe dream back in 2012. It is now a reality 7 years later. Why did this happen? It happened because of this sociology. We did not try to do this on our own. NCATS didn't within NIH, and NIH didn't do it within the ecosystem. We involved our partners from FDA from the very beginning. They've been members of the project team from the very beginning. We work with DARPA, who a lot of you know, to make this happen. A number of pharmaceutical companies and the IQ Consortium, which is a pharma consortium, and through those developed these microfluidic platforms for 12 different human organs and tied them together in different combinations. And this is what they look like. There's just one platform from a company called Ambulate, but there's many different versions. And for the scientists, otherwise known as, we're lovingly known as geeks, for those of you who, it's okay to call us that. We like that. These are the things which are now possible with these kinds of platforms that were never possible before. The things that happen within a mouse, but the mouse is not a human, and that's the problem. But you can do this now with these novel technologies and keep these alive and representing human tissues in a very accurate way. And then what we've started to do is to model human diseases on these. So if you could take normal iPS cells, could you take them from people with diseases and test diseases? Instead of making a mouse, make a chip platform and use that to study human physiology and disease. Turns out to be incredibly valuable. So again, what I'm getting at here is that it is possible, and these are some of the things that we're doing, and the other, I just use NCAS examples, there are others at other institutes, but the point is that what we have to focus on is maintaining the ecosystem to have this happen. And everything I've shown you has been a collaboration between ourselves and regulatory agencies, pharma and biotech, and or patient organizations. Everything, all of those. None of these have we done on our own. I just want to close with two things that I think might be useful for the discussion. You might, in case you don't hear about these later. There's a part of the President's management agenda to, as I said, to improve transfer of federally funded technologies from lab to market. That's a lot of what we're talking about here. If you haven't looked at this, please do. There's a lot of really great material in this document about what this group of people has thought about. If you just look at the words here and what they recommend is increasing entrepreneurship, increasing the transfer from public to private sector, increasing public and private collaborations, and increasing the efficiency by which technologies get out of the NIH space and into the private sector space. And this is being run by Walter Kopan at NIST, but it's really, as you can see, these are all the different agencies that your tax dollars support. This is HHS here, and NIH is a little organelle within HHS, but there's lots of other agencies focusing on all these different problems, including BIDOL, tech transfer, improving lab to market, SBIR, small business, the I-Corps program from NSF, and this consortium for tech transfer. I thought I should tell you this because I found I was talking to somebody the other day on the phone, and I realized that this happened so long ago that this person who's very experienced didn't know about this. So I thought, and it's very, very well known within NIH, so I thought you should know because I thought it would be pertinent to the discussion we may have later on. And I'm just going to read this because it's important. It's important to get the language right. In the late 80s, this issue came up that we're discussing, the role of NIH in drug prices and concern about prices of drugs. That the NIH included what was called a reasonable pricing clause in its licenses to NIH inventions, including those arising from CRADAs, as a cooperative research and development agreement. And what the language said was, if the CRADA resulted in an invention and the participating company wanted to take an exclusive license, which they always do, to that invention, then the company would be required to reasonably, quote unquote, reasonably price the commercialized drug that resulted from the invention, if one did result years later and you got through the valley of death. So the clause only applied to licenses of NIH inventions. This was not applied to extramural, to academic funded organizations. This was NIH intramural, about 10% of the budget only. But it had, and the official word here is from the Office of the Science Policy, so this is not my word, it's their word, chilling effect on NIH and industry collaborations. In 1994, a long time ago, NIH held public hearings to consider whether this pricing clause was promoting the NIH mission to enhance the health of the American people or was constraining it. And the consensus was that the use of the clause was driving industry away from collaborations, resulting in fewer opportunities to advance research and promising treatments. As a result, NIH removed the reasonable pricing clause from the CRADA agreements and its exclusive licenses in 1995 to overcome this roadblock to collaborations. And once the clause was removed, the number of new collaborations tripled over the next couple of years. So just keep that in mind. A lot of people don't realize that you know, as is often said, nothing new under the sun. But this is a long time ago, and I find that as a result, a lot of people may not know about this history. Okay, so lastly, a philosophical point, and this is a point I make with every group because it's essential to translation. Translation is an obligatorily team sport. Those of you who were trained in science probably were trained to work only on your own. I certainly was. So what is a team? A team requires top performers with a wide variety of different expertise to work together to a goal that none of them could do on their own. Now, when I was in training, this was not what I was taught. What I was taught is I wanted to be this person. So, who is this person? Grainy black and white photograph, guy in a white coat holding out a petri dish with crud on it. That has got to be Fleming, Alexander Fleming, who famously did not clean up his bench before he went on vacation, came back and there was mold growing on his bench and it turned out to be penicillin. So this is the idea of the lone scientist working by candlelight at night and eureka moment. That is how basic research works. I've had that experience. It is intoxicating. And it's why people stay in science, despite all of the very rational reasons to go do something else. This is why people stay in science. That works for fundamental science. I sometimes talk about this as science is golf. You know, it's a solitary exercise, maybe with your caddy, you know, your lab head, but that is not translation. Translation is football. And if you think about it, I realize we're off season here, but I hope you can think about this. Or think about soccer too, if you're like soccer better. It's the same issue. Think about the defense here. I just showed you the defense. The defense is Mother Nature, who does not give up her secrets willingly. This is a very strong defense. The only reason this team scores is that these team members are completely relying on each other and handing off from one to the other. They're not hogging the ball. They're not saying the credit's all mine. They're all working together to score a touchdown. If that doesn't happen, teams don't score. And that is the case in translation as well. And so I'll just leave you with this vision. I really like this. I don't know if everybody's ever played this, but it's something that I was forced to do in one of these team building exercises, and it's actually quite useful. What it is you take about 12 people and you give them a rope that's tied into a circle, and they have to squat down and pull on it. And for them to stay upright, they all have to be very sensitive about the needs of their collaborators and all pull the same way. If one pulls a little bit too much, the whole team falls down. Or if it doesn't pull enough, the whole team falls down and the whole exercise goes away. And this is really how this ecosystem works at its best. So my take-home message, innovation, partnership, mutual recognition of the complementary contributions of many team members and different organizations are absolutely required to advance science and medicine to benefit the millions of patients who are in need and need us to succeed. So I'm really excited about this meeting happening, and I'm looking forward to the discussion for the next day. I have a number of colleagues here who will be here tomorrow. I can't be here tomorrow, but I look forward to the discussion, and I want to thank Senator Bingaman and the Academy for having this workshop on this extremely important topic. So thank you. I'm tempted to answer that in an unusual way, but yes, of course. Yeah.
My name is Amitabh Chandra. I'm a member of the committee. You spent a lot of time, you did an awesome job of helping us understand the valley of death.
Yep.
What your good office does
Yep.
in overcoming that.
Yep.
I'm thinking about a different challenge.
Yep.
Which is the challenge of understanding. Dr. Austin did a terrific job of talking about the valley of death and sort of what we can do to bring new drugs to market by shortening that valley, by
Yep.
removing it. I'm thinking about a different challenge, which is understanding the biology behind why do existing drugs fail?
Yeah.
In humans. So they work initially, and then there is some, there's some target alteration or DNA damage, and I become resistant to the drug I'm on.
Yep.
It seems like that is a piece of this
Oh, it is.
puzzle.
Oh, it
And
is.
I was trying to understand, what does your office do to help us understand that piece of human biology?
Yeah, we do a lot of that, particularly AID and NCI do a lot of that, given the nature of the work that they do. And so I didn't obviously have time to talk about everything we do, but we have a very large program taking every drug ever approved for human use and testing it in many, 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 many different assays and making all those data public. And one of the things that we do is to try to look at variations in individuals and say, if this person responds and this person doesn't, why? And we're doing a lot of that with the NCI. Christine Colvis, who's sitting back there, will also talk about some of this on the repurposing issue, which is a little bit different. So there's an example of a drug that continues working and might work in a different disease, and how do you identify those? But it's, yes, that is a translational problem too, yeah. Yeah, anybody? Yeah,
Hey.
yeah,
Sir.
sure.
Ross Anderson, CAREM, CEO CAREM, geospatial epidemiology company. What about Mother Nature and the Big Six? Mother Nature is always the defense. So if you were on the defense, had an interception, ran it for a score, that's a team effort too. Just a thought.
And, sorry, I was having difficulty. Could somebody tell me what the? I couldn't
Sorry,
hear.
is it?
Yeah, it's not working too well.
It's working.
Oh,
Okay.
that's better.
Okay.
Yeah,
All
that's
right.
better.
All right. So what about a pick six on team football? You guys are the defense. You run it back for the score, rather than saying you're always on the offense.
So what would the?
What about an interception running back for a TD? Just asking.
Yeah.
Yeah, you could be the defense.
Yeah. Yeah.
You could reverse Moore's law.
Yeah. Yeah. Well, that's.
Or Eram.
Yeah. Well, I think that's one of the things that we're trying to do. It's not non-trivial, but I would say we're making a little bit of headway. There's
Thank you for the excellent presentation. Is this working?
Yeah.
Oh, great. My question is in relation to one of the slides you showed about now understanding at the mechanism or at this molecular level, the causes of almost all the key diseases today. And then you also stated that our body is one and many of the diseases are connected in terms of their onset and mechanisms, which is true. We also know now that a lot of these key chronic diseases, of course, cost a lot of money and productivity, start way early, 15 years, maybe earlier. And we can know from the molecular basis that you do start early.
Yep.
So I was wondering, in your strategy or in your policy at the NIH, how much effort is intentionally dedicated to perhaps taking an offensive measure or a preventive measure early on, because prevention is likely to get us to a better outcome and productivity and lower cost. But really start, all of us have some type of risk for all those diseases.
Right.
So can we start when we are healthy?
Yeah.
Because we know we have those risk factors and we identified them already what they are, and how do we really deal with the onset of those so we can prevent or delay the onset to much later in life?
Yeah, it's a fantastic question. And there are a number of things at NIH doing that. Probably what exemplifies them is the All of Us research program, otherwise known as the Precision Medicine Initiative, and that is recruiting we hope 1 million or more Americans who are healthy within that broad definition, and to do not only genome sequencing, but all kinds of blood tests and profiling and imaging and all those kinds of things to try to identify who's going to get these common diseases earlier in a prospective cohort. There's lots and lots and lots of studies that NIH are doing with people who are already sick, and then trying to compare those with controls, and those are great studies too, but they're not prospective, and so the All of Us program is disease agnostic. Like this, it's just taking healthy people and saying, well, gosh, if you have this genome profile or this blood profile, what diseases do you get? And do you get both hypertension and diabetes, or you get diabetes and ALS, or do you get this and that? And we hope that will help us shed light on the fact that individual genetic differences do either protect from or predispose to more than one condition and disease. Yeah, and so that's now it's about 1.5 years into the program. It has about 250,000 people recruited already. If all of you haven't signed up, you can go to allofus.gov and you can sign up from there, and you get all your data back too, which is another advantage. Yeah. Any other? Yeah.
It seems to me that fairly recently drug development for HIV and for hepatitis C have done a pretty good job about reducing the value of death.
Yep.
Are there other areas such as those that have a greater success at doing those things, and what can we learn from them? How can we apply those lessons elsewhere?
Yeah. So I'll give you an answer from the standpoint of a neurologist not from an infectious disease doc. But I tend to think about infectious diseases as a little bit easier because they are an outside organism that don't belong there. So you can, sometimes the drugs you can use are they're just different enough that they don't have off-target effects. They don't have side effects. So it's an unusual case. But I think the other reason this happened was a long investment in understanding the virology, the basic fundamental virology of HIV, and even more than that, hepatitis B, C, and B before it, and A, so that we understood everything about the life cycle of this organism, so we knew where to intervene. The other lesson, I think particularly from the HIV world, those of you may remember that this was the result of a very large collaboration among a number of pharmaceutical companies who all came together at the time and shared their data. Was rather, at the time, atypical behavior, and it led to much more rapid success. And I think that has led to lots of other collaborations, both within pharma and between pharma and the NIH, things like the Accelerating Medicines Partnership, which is a $250 million project each between a number of pharmaceutical companies and the NIH on lupus, rheumatoid arthritis, type 2 diabetes, and Alzheimer's disease. And there's a new one on cancer and a new one on Parkinson's disease as well, taking the resources of both in this model, this collaborative model. The other area, which is again a special case, is the area of rare diseases. Sometimes those can be more straightforward because the cause of them is more straightforward. It's a single gene disease as opposed to diabetes, which is probably a disease of hundreds or thousands of genes. And so we're very actively taking those lessons from both rare diseases and infectious diseases and applying them to the really hard cases like neuropsychiatric diseases, and neurological diseases, which are really the hardest. Yeah.
I guess that'd just be the last question.
Yeah, just one last question if there's, yeah.
Thanks. Bob Sampat, Columbia University. I'm not sure if this is working. So excellent presentation, thank you. I thought that the whole point of the Scannell article on Room's Law was precisely that we don't want to kind of science the heck out of this. So he calls out kind of what he calls the brute force basic research bias or molecular kind of reductionism, the idea that all advances stem from genomics and the only hard problem is to sort of quote unquote translate them.
Yeah.
I'm a mere economist and don't know anything, but I'd be curious on your perspective on that.
Yeah. So what I mean by the sciencing the heck out of it is the application of the scientific paradigm to the causes of failure in the translational space, and to view all of the steps that go into making a therapeutic, some of which are really quite arcane, like figuring out how to do a salt form or lyophilize your compound. Now that may sound really boring and dumb, but that can have a fundamental effect on whether the drug gets absorbed into the body that you made. And if you don't do it right, your drug doesn't work. Now, we really don't understand how to do that very well. There is a lot of material science and fundamental science in that space which has just never been supported as an academic discipline before. And so and another way to think about this is that, you know, we think that the most interesting science is actually being done to be done in the most complex system, and the most complex system is the human, not a fruit fly or not a worm. I love worms. I love flies. I've published a lot of papers on these, but the degree of difficulty is much greater in the human, and yet it's very hard to get funding to at least traditionally it has been, to do this kind of work in humans, and so that's the kind of science that we're supporting. So okay, good.
We have one more who
One more, Doctor. Thank you for your work, and thank you for your remarks. Is this on? Can you hear me?
Just
All right. Let's see. How's that?
Good.
So I just have a question about the math on
Yeah.
Ebrum's law. When you look at R&D costs, do you look at actual R&D costs, or do you look at reported R&D costs that frequently include acquisitions by drug companies of smaller
Yeah.
companies. For example,
Yeah.
when the drug that came called
Yep.
was transferred from Kite to Gilead with 2 NIH patents.
Yep.
The Kite had reported $321 million of R&D
Yep.
to the SEC right up to the moment of that acquisition, which then
Yep.
cost $11.9
Yep.
billion,
Right.
I think, or 8.7. I think it was 11.9.
Yep.
So when eRoom's law is calculated, is it looking at actual R&D costs, or is it looking at costs that companies sometimes report as R&D,
Yeah.
but are actually buying a drug based on what they perceive to be the
Yeah,
future
it's a-
value they can sell?
Yeah,
it
it's a really good question. So as far as the ones that we use the same ones that everybody uses, mainly the Tufts ones, the ones that Joe DeMasi makes, and we're actually doing the forum is actually doing a big program on late stage failure with the Tufts people right now. The eRoom's law, if you look at when eRoom's law, the paper was written, it was written in 2012, and this looks back, so it ends in 2010 and goes back to 1950. So this phenomenon of acquisition, for the most part, was pretty rare up until really the 90s, 2000s. So my guess is that most of it is not that phenomenon. It's internal drug development in companies.
But it's derived from Joe DeMasi's work at Tufts.
The eRoom's law, I'd have to go back. You can go back and look at the paper too. I don't remember right offhand what the data were that Jack Scannell used. Does anybody remember? He's the senior author on the paper.
I'll look.
Yeah.
It can't be Tufts because Tufts doesn't go back to 1950s.
Yeah. Yep. Yep.
Totally what?
Total industry spending on R&D,
Okay.
just divided by number of products that come out of the pipeline.
Yep. Yeah. So it's an aggregate.
I'll go back and look.
So that would include that. Yeah.
And so maybe things don't look as dire as
Yep. Yep. Yep.
Great. Thank you.
Okay. Thank you.